Hello everyone, I hope you all are doing really well. So today we'll be studying about plant disease control measures, okay. So we know that there are many majorly fungus causes diseases in plants, and there are many fungicides and many chemicals we use to get rid of or prevent these diseases. So basically we can just classify the methods into three basic things, okay. First one will be preventive measures. Preventive means we prevent the pathogen from entering our field. Second one will be curative. The pathogen has entered, but we control it afterwards. And immunization means we sow such seeds that are immune to or resistant to such so and so pathogens, okay. So this topic is really very important for us. Just before we start about chemical fungicides, etc., we must be knowing that there are many other methods that actually form the very basis of controlling these diseases. If we skip those steps, then no matter how many fungicides we use, how many chemicals we use, our farms will definitely destroy sooner or later, okay. So first of all, we prevent diseases. For example, in human body, we prevent diseases by eating hygienic food, by avoiding stale food, and then by exercising, by avoiding going to places with high pollution, okay. So similar are the measures with the plants also, okay. When the corona time came, we were avoiding the viruses by hand sanitization, by using mask. So we just prevent it from entering itself into our body. After it enters, then there goes on a treatment, and after a pathogen has entered, that will definitely cause negative effects in one's body, okay. So firstly we'll be talking about preventive measures. This is also called prophylactic, okay. So what is prophylaxis? Prevention is better than cure. This is the basic meaning of this word, okay. Preventive measures that are taken to avoid the introduction, establishment, or spread of a pathogen in crops. This is called prophylaxis. Historically, when we open Arun Katyan, even the curative measures are given under prophylaxis, because historically, before the epidemic spread level occurred, before that, whatever measures were being used even to control after infection also, they were all being called prophylaxis. But in verbal meaning, prophylaxis mean simply that we cut off the pathogen, or we just create a barrier in the very first stage, that it does not even cause infection to our plant, okay. So we'll be studying prophylaxis according to its true verbal sense only. So first part of prophylaxis, first method contains exclusion. Exclusion, as the word suggests, we just simply prevent the entry or introduction of pathogen into a disease-free area. The area which is already disease-free, how will a disease enter over there? The pathogen will be carried through some seeds, because when a new season comes, we sow some seeds, or maybe the tools that we are using over there, they are infected with some pathogens, okay. Or maybe we are not taking care of the plant when it is in its growth stage or seed forming stage, and the seeds are already new to that area. So maybe when a new seed variety is sown into a new area, that develops some physiological disorder, or basically some kind of pathogen or disease which was not diagnosed earlier, okay. And the very first step will be quarantine. Although quarantine, as you all know, this is the very known word now after corona. Quarantine basically means whatever new variety of seeds will be coming to a place, will be imported into our area, that a variety will be quarantined for some time. That will be checked for some pathogens or diseases or weeds if it is carrying, and maybe there are some new diseases that are not even known to that area, and if we sow the seeds over there, the area will get prone to that disease, okay. So we basically quarantine the new varieties in the start itself, so they don't carry any new diseases to our local place where they are being introduced, okay. So yep, quarantine is basically a regulatory measure This is done by the government and it is a legal restriction. Plant Quarantine Act of 2003 is the example, which was passed under Destructive Insects and Pests Act of 1914, okay. And the example over here is like bunchy top of banana or citrus canker. These are the diseases that can get introduced with the seed plants or seeds of banana or citrus, okay. And there are many more examples. We will go deep into this topic in upcoming videos. This specific lecture is only an introduction, because if we go on doing everything together, first of all we lose track of the classification itself. It becomes very complex, okay. And secondly, the video becomes very big. So I just want to keep it short and easier to remember. So firstly, in this video today we will be understanding the basic differences among the terminology of this topic, and in the next video we will be understanding these things with much more practical examples that are carried out in our fields themselves, okay. Now comes the direct protection. Direct protection is also part of prophylaxis. How is it the part, okay? Basically, this is the definition you can read, that when we directly avoid the infection near the plant, how will we do that, okay? Suppose a pathogen has entered an area, we can still control it. How? When it reaches to our plants, we use some protective measures over there, or we can just control some environmental factors so that that pathogen doesn't get some any of the conducive conditions to grow in that field itself. So protecting the plant directly from infection either by applying chemicals or by modifying environment. First will be the chemical prophylaxis. How will it be done? Spraying or dusting of protectant fungicides. Sulfur, copper, mancozeb, etc. are the examples of such fungicides which we either spray over the plant or dust, and that basically prevents the infection from occurring. You need to know that wherever the prophylaxis word comes, we are talking about that the infection has not yet occurred in the plant. Maybe it is around the plant, but it has not entered the plant yet, okay. So we spray or dust the plant with some chemicals like sulfur, copper, mancozeb, etc. So the infection doesn't enter, and at the contact itself it gets killed, okay. So they do not kill the fungus inside the plant, rather they prevent a new infection from occurring into the plant. How? Most of the chemical fungicides that we use, they are contact fungicides. That means they do their action right before the fungus has caused or entered the plant, okay. They act before infection occurs by forming a protective barrier on plant surfaces. Hence their main role is preventive and not curative. This we need to understand. Wherever the word comes contact fungicide or preventive or contact or preventive fungicide, that simply means that the infection has not yet occurred. The infection, when it comes in contact to the plant will be having that dusted or sprayed particles of a chemical, and that chemical will prevent the infection from entering into the plant by killing it before it enters, okay. So sulfur, copper compounds, these are the examples, as I already told. Mancozeb, Xenum, Kaptan, etc. are also the examples, and Bodex mixture is an example of copper compounds, and sulfur fungicides are basically used against powdery mildew. These are very important points, but we will be going in depth in upcoming videos, okay. So environmental manipulation is the second thing. Environmental manipulation is the second prophylaxis that can be used. If we can use the chemical procedures also to prevent the infection, and we can just control the environment itself, maybe in the greenhouse or warehouses also, wherever the seed has been stored, okay. Then altering the time of sowing and harvesting also. Suppose we sow a crop at a time when a specific pathogen has very high chances of surviving, because that is the environment in which it easily survives. Or maybe we harvest a little bit later, and the crop gets highly susceptible to get infected in that particular climate type also. So what we do is we alter our sowing and harvesting time just to escape the attack of that particular pathogen. So this is environmental manipulation for that crop. So altering the time of sowing harvesting in order to make it favorable for host. For host it is favorable, and unfavorable for the pathogen. protecting plants from cold, frost, heat, etc., because obviously physical injuries make them more susceptible to other kinds of diseases and insect pests also. Okay, so we basically protect them from such injuries also. And maintaining nutrient elements also. You must be knowing that if a plant gets too much of nutrients or very less of some nutrients, they develop some physiological disorders, and some physiological disorders are so that are accompanied by some specific diseases in plants. We'll be discussing such things in upcoming videos as we go through specific crops also. So basically we need to know that this is a very important step to be taken, and whenever we read a package of practices for any crop, these are the basic things that are kept in mind while deciding the sowing and harvesting times of different crops. Okay, and the third one is the eradication. Once the pathogen has been introduced, okay, the aim is to eliminate it now or destroy it after it has been introduced on host and before it spreads further. Okay, so that the spread can be blocked, or a barrier can be created that it doesn't spread further. Then there are some methods to do that also. Crop rotation, first of all. Suppose an infection has been introduced in a field. So what we'll do is we'll rotate that field with different non-host crops of that infection itself. Okay, so maybe the diseases that occur in paddy doesn't occur in cotton. So maybe we can skip sowing paddy for this season or sow cotton, or maybe we can skip sowing cotton in that field and sow paddy according to the season over there. So this is how we avoid diseases for a specific host. Suppose one specific disease has occurred over our field, and that particular disease or that particular pathogen survives for 2 to 3 months or maybe 6 to 7 months, and after that it dies itself if it doesn't get a host. Okay, so we'll smartly not introduce the host itself into that field so that it doesn't it doesn't get any host, and doesn't survive only. So after a few seasons, 1 or 2 seasons, we can reintroduce our crop that is the host. Okay, so rotating with non-host crops breaks the life cycle of soil-borne pathogens. Soil-borne basically, because the pathogens will survive in various things, but crop rotation basically helps in soil-borne pathogens. There can be some weeds also that can act as host for a specific diseases, and then we'll be eliminating those also, that alternate hosts also. Okay, there are some weeds of maybe wheat that are the host for the same diseases as the wheat is. So we will eliminate those also. Then sanitation also, we'll remove any plant debris, weeds or residues, whatever extra things are there in the unneeded things are there in the plant, we'll remove in the field, sorry, we'll remove those also. And then comes the chemical eradication. Soil fumigation or chemical treatment to destroy resting spores can be given. We'll definitely go into depth. Right now we are just getting used to or known to some basic terminologies. Systemic or curative fungicides are exception to chemical prophylaxis. As I already told, that prophylaxis means preventive measures. Okay, and now we come to systemic or curative. So wherever the term systemic comes or curative comes, that means the infection has occurred now. The infection or pathogen has entered into the plant, and now it needs to be controlled over there. So what will the systemic, as the word suggests, what will the systemic chemicals or fungicides do? They will enter the plant also, just as the pathogen has entered. They will also enter the plant, and they will enter the pathogen then, and destroy the basic functioning of pathogen. Some systemic fungicides like carbendazim, triazoles, metalaxyl can enter plant tissues and suppress the fungus even after infection. Okay, they can enter the plant tissues. Such fungicides are curative or eradicative in action because they eliminate the established pathogen. They just kill the pathogens that has been already introduced into the crop. And the third measure comes as immunization or host resistance. What is that? We have already discussed that sowing of resistant varieties. The seeds that are being introduced into the field are resistant, and such immunity or resistance can help in plant disease control measures. So breeding resistant varieties through selection and hybridization, these are the methods of breeding. And then there can also be temporary immunization. Among temporary immunization, one thing that comes is chemotherapy. Chemo is simply, what is chemotherapy? Chemo simply means chemical and therapy means treatment. So chemical treatment that we give to our plants to get rid of such pathogens. So treatment of diseased plants by the use of chemicals that move inside the plant system and inhibit or kill the pathogen after infection has already occurred. Okay. Then chemical fungicides or antibiotics are absorbed through root systems or leaves. This is the first thing that happens. Whatever the fungicide we have introduced, that gets absorbed through roots, stems or leaves. Second thing happens is translocation in the plants via xylem and phloem. And third thing, when the chemical has gotten translocated equally among all the tissues of the plant, then they interfere with the processes of the pathogen, which was disturbing the natural processes of the plant, and they kill the pathogen. Okay, so what are the routes of application of such chemotherapy? This can be foliar spray or injection for systemic fungicides or insecticides. This is understood. I think there is no need to explain this. Then soil drenching can be done for root or wilt diseases. Obviously, the diseases that are caused through soil or are soil borne can be controlled by drenching the soil with some chemicals. Seed or tuber treatment can be done for internal pathogens, for internally seed borne pathogens. And then fumigation or vapor treatment can be done for stored produce or greenhouse, so that when we store the seeds that are to be sown, they do not carry any pathogen, etc. So fumigation or vapor treatment can be very helpful. So that is it for today. Thank you so much. I hope this video helped you, and in next videos we'll be discussing the same things in much more detail and depth. Thank you.